I'm Jamie Angus, and I'm mom to Oliver, who's 16, who has TANGO2. And I'm grateful that this is my second conference, and I'm loving meeting some of the newer families too, and just appreciate this opportunity. And I'm introducing Dr. Lina Gonzalez. She's an assistant professor of pediatrics in the Division of Genetics and Medical Genomics at University of Pittsburgh. She received her degree in medicine from the University of Aleppo School of Medicine in Aleppo, Syria. She is board certified in internal medicine, medical genetics and genomics, and medical biochemical genetics. Dr. Gonzalez has been awarded a National Institutes of Health K08 Mentored Clinical Scientific Research Career Development Award under National Human Genome Research Institute in 2019 for her grant Precision Genomic Medicine in the Plain Communities and its impact on the plain and general population. She also has been funded for 1 year from the TANGO2 Research Foundation in 2020. Dr. Gonzalez's focus is on the genetic disorder in the Plain people, Amish and Mennonites in Western Pennsylvania, to characterize novel genetic disorders or novel mutations. She also has interest in studying the pathophysiology of TANGO2 deficiency. Welcome, Dr. Gonzalez.
Okay, so I recognize that my talk might be a little bit hard to understand. I'm going to try to simplify as much as possible, but feel free to interrupt me if if it's too much. So as you can see here, the title Mitochondrial Dysfunction in TANGO2-Related Disorder and What is Next. As you heard, one of my grants, the K08 grant, is focused on the Plain community, Amish and Mennonite, but also I'm interested with the TANGO2 pathophysiology. And interestingly, also the Amish have TANGO2 deficiency with the common deletion, the 3 to 9 exon deletion. So it is in both the general population and in the Amish, which is, fulfills the purpose of both my research interest. So as background, you already heard 2 talks about a lot of background about TANGO2. So it stands for the Transport and Golgi Organization 2 homolog. We still don't know the exact function of the TANGO2, but we are working on it. Here in my talk, I'm going to focus mostly on the mitochondria and how it is affected. And if you think, probably all of you by now know that mitochondria is the powerhouse of the cell, so we get the energy mostly from the mitochondria. Before doing any research study, one of the important things to do is to, for example, I'm doing my research experiments on the skin fibroblasts. So one of the main things to see whether this gene is expressed in the skin fibroblast. So all our cells have all the genes, but genes get translated to proteins in some tissues. For TANGO2, you can see that it is expressed in most of the tissues in the human body, and that's actually it reflects the clinical manifestations that we see in these patients because they have many organs affected. So as you can see here, I don't know if the pointer here. So this is the skin, and you can see that TANGO2 is expressed in the skin fibroblast. So that tells me, well, I can go ahead and do my studies on the skin fibroblast. What do we know about the pathophysiology or the mechanism of TANGO2? We really don't know much, but we are making progress. So why are we studying mitochondria in the TANGO2 deficiency? Many of you probably have been told with your kids when they were little that they have mitochondrial disease. There are several organs affected with patients with TANGO2 deficiency, and we see this in patients with mitochondrial disorders. There is also a study that found the TANGO2 gene in mice is expressed in mitochondria in big quantities in the brain. There are some labs that have been seen in these patients that we see in general in patients with mitochondrial diseases. And I know this is complicated, but probably if you've seen neurologists or geneticists, you've heard about acylcarnitine profile. And acylcarnitine profile looks for different things, but some of it is fat metabolism which happens in the mitochondria. Patients have episodic rhabdomyolysis, which we can see it with some of the disorders in fat metabolism, like namely VLCAD deficiency, which is the fatty acid disorder. Hypoglycemia, which is low glucose, we can see it in mitochondrial disorder. Hyperammonemia, we can see it also in some mitochondrial disorder. Urea cycle disorders, which is part of the urea cycle is in the mitochondria. And severe metabolic acidosis. So all of this that we see in mitochondrial disease. There are some of the patients we see, they have clinical studies on the tissues where they look at the different complexes. There are 5 mitochondrial complexes in the mitochondria, and there are some abnormalities in these complexes. So by looking at all of these, it seems that there are several networks affected, and many of these networks are inside of the mitochondria. So this is why there were several studies done looking at mitochondria specifically. There are discrepancies, for sure, when we talk about mitochondria involvement in TANGO2 deficiency. So there are studies done by our colleagues, and one of them was done in skin fibroblast, and they have similar findings to what my lab did, where they looked at mitochondrial morphology, the size and number, and they found that mitochondria is smaller in size and increased in number, and we can talk more about what that means. And they also found that TANGO2 is partly localized in mitochondria. When we say partly localized in mitochondria, we're talking about maybe it's not all in there, it's expressed somewhere else, maybe in the cytoplasm, other organelles, we don't know exactly where. There is also another study which was done on primary myoblasts, which is cells from muscle, and they did not find change in the mitochondrial size and number. They also look at something called oxygen consumption rate, which is a function that we can measure in the mitochondria, like measuring the oxygen, and they found that these patients had normal oxygen consumption rate. So they were these studies. My lab was already doing studies, so we really wanted to know, is it affected or it's not affected? We know on the clinical side, we're seeing all these abnormal labs that tells us there's something wrong with the mitochondria, but we also have these studies, so we continued with our. These are the skin fibroblasts were from 3 different patients. P1 and P2 are patients at UPMC, so they were our patients. They are siblings. I never got to meet patient P2 because he was deceased when he was 6 years ago, and that was more than 15 years ago, so that was before I started my fellowship in genetics. Both P1 and P2 have homozygous deletion for exon 3 to 9, the common deletion, and they were both severely affected with severe developmental delay, failure to thrive. P1, I still follow her. She's 16 years of age now. She has severe intractable seizures, difficulty swallowing, and low muscle tone. And on the MRI, the brain, there is some volume loss in the brain. I don't have much information about P2 except that he had the profound delay, but also had severe lactic acidosis. It's interesting to see how both these siblings, they have the same deletion. They were both severely affected with the developmental delay, the hypotonia, but P1 who still follow never had lactic acidosis. She never had acidosis in the blood, while her brother died with severe lactic acidosis. P3 is a patient of a collaborator who patient have 2 different mutations. One of them is the common deletion of exon 3 to 9, and the other one is also a relatively common splice, we call it splice site mutation. So with all these mutations, you don't expect a protein to be translated. The one that Dr. McKenzie was talking about, how we go from DNA to mRNA to protein. So, but with these deletions, we don't expect any protein to be translated. P3 actually had only 1 episode of rhabdomyolysis, lactic acidosis, and low glucose hypoglycemia. She had mild global developmental delay, and she's much less severely affected than the other 2. I actually don't know if this is the daughter of someone here in the, maybe, because I don't know this patient. So, first thing we wanted really to see is TANGO2 in the mitochondria or not. So what you see here, this is a whole cell lysate. So what we do is take cells from these patients, lyse them, like smash them, and then take a fluid which has the protein, load them on what we call a gel, and look at bands. So wild type is controlled cell line, so that's healthy individual, and then P1, P2, and P3, these are the patients. So with the TANGO2, so we're looking at the TANGO2 protein. So this is the normal band that we would like to see. And it is present in the control while we don't see it in the patient. So it's completely absent there. GAPDH, this is just a control gene just to tell us that we did the experiment well. The other portion of this is, so this part is taking the whole, everything is in the cell, all the proteins in the cell. This part here is just isolating the mitochondria and then looking at the protein in the mitochondria. So what we found is, yes, there is a band of TANGO2 in the control. It is less intense than the other one that we see with the whole cell lysate, but it tells us that TANGO2 is at least partially in the mitochondria. Of course, it's not in the patients. And beta-actin here is also another control protein, just to tell us that we did the experiment well. This is a little bit complicated, but we'll try to simplify it. So one of the things we look at is mitochondrial respiration. So mitochondria get into different reactions and consumes oxygen. And by doing that, it generates energy in the form of ATP. There are different forms to measure that. One of the first one is called basal respiration. And basal respiration is measuring how much oxygen the mitochondria is consuming, but that's at rest. Maximal respiration is maximal respiration is when we're measuring oxygen consumption from the mitochondria at maximal capacity, and that's when we are patients, for example, are sick or they're working hard, so the mitochondria have to work very hard. So there is at rest and there is maximal respiration. Then there is a spare capacity, and spare capacity looks for the difference between the basal respiration and maximal respiration. So we did the study under 2 different conditions. One of them, we do this in a 96, in a plate, small like this, and we put it in an instrument called Seahorse. We do the study in 2 different conditions, having the cells with media, nutrients, one of them with glucose, and that these are the normal conditions that we have the cells with. And that means we have the cells with no stress at all. And then the other condition is without glucose, and that is by itself stressful for the cells. So So when we don't have glucose, we want the mitochondria should work really harder. And that is similar to what happens during illness. So we wanted to see the differences. This is a graph. So this graph here, this is what you see, you know, in the protocol. And I know this is complicated, but just we can look at few things. So when we are looking at basal respiration, I keep missing the, here it is. So basal respiration, we're looking at this line here and do the measurement from here to there. So that's basal respiration, how good the mitochondria is doing during rest. Then with the maximal respiration, we do this measurement here from the top to the bottom. And then the spare capacity is, should keep, the spare capacity is the difference between the maximal respiration and the basal respiration. So we're looking at this difference. So here, this figure is with glucose. So wild type, this is in blue. P1 is the patient that is 16 years old now. P2 is the one, the most severe that was deceased currently. And P3 is the less severe. I don't know, does this work, Mike? Or no, this is some, yeah? How do I do the pointer?
The red button.
Okay, this is easier. So wild type, so you can see here the wild type, the blue, and P3, which is in purple. You can see how they have very similar, they're overlaying on top of each other. So that tells us that P3 is doing really well. You know, when the skin fibroblasts are not under stress. With the P1, you can see here that it is the basal respiration is lower, the maximal respiration is lower, and of course the difference is also lower. With the P2 is the lowest for basal and maximal respiration. So that is under glucose with, you know, no stress. Without glucose here or with stress, we can see that, you know, P1 and P2, they are even lower, but also P3 here, and that's the patient that is doing well in general, under stress is not doing that well. So it's also less compared with the wild type for basal respiration and maximal respiration. This is the same graph as the previous one, but it has a statistics. These little stars that you see here, it just tells us that these are statistically significant. And again, C is the control, P3 is the patient, and here it's divided as basal respiration, maximal, and spare capacity. This is with glucose. Some people like this version more than the other one. But you can see how P1, P2 here is less for basal, it is less for maximal, and it is less for spare capacity, while for P3 is very similar to the control. And that's with glucose. Without glucose, all of them are lower than the control. So as a conclusion with this, so with glucose, so basal respiration at rest and maximum and spare capacity were similar between control and P3, while the least affected, while the other two were lower, while with no glucose, all of them were actually lower than the control. This takes us to the other part that is done in the mitochondria. We look at the ATP production. So ATP is the energy that is produced in the cells, and it is important for the cells to work. It comes from two different parts. It can come from glycolysis, which is a metabolism from glucose, but also from the mitochondria. The majority of the ATP comes from the mitochondria. So ATP is important for the cell function. because we're looking at the mitochondria, and the majority of ATP comes from the mitochondria, we wanted to study this further. We did also this experiment in the same instrument called the seahorse, and what we looked here is at ATP production. The experiment by itself, we can do different measurements, ATP that's coming from glucose or glycolysis, ATP that's coming from mitochondria, and then you can put both of them together. And as you can see here, with glucose, these are all with glucose. So this is the control, these are the patients. They are actually producing more ATP from glucose than the control. So, which I wasn't expecting that, so we did repeat it, of course, several times, and this pattern was consistent. From mitochondria, ATP production with glucose, so we did see a decrease in P1 and P2. P3 was similar to control. So they were P3 was able to generate enough ATP from mitochondria without stress, while with P1 and P2, they had less ATP produced from the mitochondria. If we put these together, the patients during, with no illness or with no stress when under glucose, they are doing pretty good. If you take mitochondrial and glycolytic, so this is the total ATP, and they are kind of similar to the control. So this is with glucose. When we looked at it without glucose, and here I am combining both. This is a little bit busy, so we're going to go through it. So the green represent with glucose, and red with no glucose. So if we're looking here at the glycolytic ATP production, we see that without glucose, the ATP produced from the glycolysis is decreased, and that's expected because there's no glucose. So how are you going to get ATP? So this is expected result. Mitochondrial ATP production, what we've seen, and that was to our surprise, is with no glucose, mitochondria are really trying to work hard. Yes, there is some dysfunction that we saw with the oxygen consumption rate, but then under stress with no glucose, this is controlled, so this is expected. But these are the patients, P1, P2, P3. P3, we didn't see much difference because anyway they were doing good. But they are working harder and they are increasing the ATP production. This was also confirmed with multiple times doing it. And when you look at the total ATP production, and that was a big surprise actually for us, said, well, the cells are producing ATP. They're kind of, when we're taking it both together, so they're pretty similar actually to the control. So here it was a little bit puzzling, but to look more into details about that, we looked at different, this is the same summary. What we looked at the ATP in a different way. This is a different assay. This is the same assay that I just showed you, ATP production with glucose, and they are similar. This is the total ATP. We did a different experiment called ATP light assay. It's totally different in different instrument, and this measures how much ATP, net ATP or left ATP is in the cells. So if you look here, all patients, even P3 with glucose, they have less net ATP or leftover ATP in the cells. So this tells us that TANGO2 cells, yes, they have some mitochondrial dysfunction, but they are really, when you put them under stress, they try to work hard, generate ATP, but they really consume their ATP and they use their ATP more or faster than the control. This is why we're seeing this here. So that was the other piece, complementary to the other one. This is the summary. The other part of mitochondria we looked at is mitochondrial dynamics. We looked at the shape and number of mitochondria and number of mitochondrial DNA. So each cell have hundreds to thousands of mitochondria. But then each mitochondria have 2 to 10 mitochondrial DNA. And the mitochondrial DNA is different than the nuclear DNA. TANGO2 is in the part of the nuclear DNA. The mitochondrial DNA, they are circular, and there is 2 to 10. So we looked at the number of this DNA in the mitochondria, and these are important for coding proteins that they are inside of the mitochondria, like the complexes that they are important for generating the ATP and other proteins. We looked at the shape and number of mitochondria. Are they circular? Are they connected with each other? The healthier mitochondria is when they are more connected to each other. We really don't want singular small mitochondria, because that's indication that it's not healthy. So we did use a protein called TOM20, and that stains the outside of the mitochondria. And if you just look at these figures here between control and the patients, you really don't see a difference. But we have a way to do the analysis, and when we looked at the analysis, we looked at the number of mitochondria per cell. So the number of mitochondria per cell in patients compared to control was bigger. And then the average volume of mitochondria was smaller. And this is, there were different studies, but I'm putting only this one. And we did also a Western blotting, looking for a specific protein that is involved with fission. And fission is when a process where mitochondria get connected with each other. So mitochondria goes into fission and fusion. Fission is when they connect with each other, and fusion when they disconnect. So there was increased fission, so they are more disconnecting from each other. So this result with these ones tells us, well, there is increased number of mitochondria, but they are smaller in size. So it is as if it is the cells are trying to compensate, like, okay, the mitochondria is not working properly, so what should we do? Let's generate more mitochondria. But the mitochondria that is generated is also not very healthy, because it's also, the shape of it is not what we like it to see connected. It's more circular and smaller in size. The other thing, as I mentioned, we were looking at the mitochondrial DNA, those circular DNA, and what we've seen here, that there wasn't much difference between mitochondrial DNA copies and the control. But if you take this result with the previous result, so there's increased number of mitochondria, but the number of mitochondrial DNA is about the same. So if you put these together, that tells us that the mitochondria does have less mitochondrial DNA. It's just because of the increased number of mitochondria. So that it doesn't mean mitochondria is empty, but they have less copies of mitochondrial DNA. So this is a summary. Number of DNA copies in each mitochondrion is decreased in patients, and number of mitochondria is increased. They are smaller in size compared to control, and this is an indication, could be a compensation for abnormal function of the mitochondria. So what does this all mean for TANGO2 child? During illness, TANGO2 child can benefit from getting more energy from IV fluids containing glucose. This is especially important when we have these patients come to the emergency room. This is why we give the emergency letter. We tell them, you know, draw these labs, start IV fluids. It's as any of the other metabolic disorders, IV glucose is not enough by itself. This is just the first step when patient comes to the emergency room, and then we add the other, usually the TPN. But this is a fast step. Okay, give him glucose while geneticists or metabolic geneticists or neurologists see the patient and start the other things that patient needs to be on. TANGO2 kids may benefit from getting mitochondrial supplements, CoQ10, levocarnitine, and or others. How much, we don't know. Carnitine usually is given when there is carnitine deficiency. So this is why if we check carnitine level and carnitine is normal, these patients don't need levocarnitine. Does CoQ10 do something or not? We're going to hear more about that from Dr. Christina. From the natural history study. But one of the things that we are trying to look is test a few compounds. That is actually the topic of my second grant that I got from the TANGO2 Foundation. There are antioxidants, and this is some of the examples, JP4, XJP. So these are compounds that they were tried in different metabolic disorders, specifically fatty acid disorders that occur in the mitochondria with lots of benefits. They are not in clinical trials or good for human use, but there are some metabolic disorders they'll benefit from it. Some of them they don't. I'm talking about on a cellular level. So this is very early. Bezafibrate is a medication that is used actually to decrease fat, decrease lipids in patients with hyperlipidemia. So it is, if it works, good, because it has been tried in other mitochondrial disorders. Elimipritide is a medication that is in clinical trial. So some of these, so this is some of what we are going to try. Some were tried on a cellular level, they're not in clinical trials. Some are in clinical trials, and some have been used for many years. Would any of these work? I don't know, but we're gonna check and see if any of these will work. But we'll continue also looking for other clues, not only mitochondria, but different pathways. This is just the many people I work with. So mainly Dr. Jerry Buckley, he's been my mentor since I was a fellow, and then, even as attending right now. But he's interested in fatty acid metabolism and mitochondrial disease. This is part of my lab and his lab. We work very closely, on day to day. So we really talk about TANGO2 a lot, especially with the lab meetings on Thursdays, and these are my current lab members. Anoki, working on the PhD students, should be focusing on the TANGO2, and Alyssa, she will be lab tech, also focusing on helping with other experiments on the TANGO2. And I'll thank everyone, and I'm happy to take any questions.